Welcome back to the Monthly Dose. I'm your host, Elaine Chang, here with my co-host, Dr. Charles Ackle.
Hi, Elaine.
Thanks for joining us. You're the Chief Medical Officer of IMI.
Yes.
And you have a huge background in cancer immunology. They call you, some may call you the OG, one of the greats. You've been around for a while.
Oh, yeah.
And the thing I want to talk about today is kind of like it's a super interesting topic. It's actually the accidental discovery of an immunotherapy for cancer. And you played a big role in that.
I stood on the shoulder of giants, as we often do. But yeah, I mean, I think you mean particularly as relates to the mycolysibacteria.
Yes, well, we
rather
have
than
IMI.
Yeah, and we're talking about IMI, of course, because there have been some huge developments in immunotherapy just in the last 10, 15 years, which is fantastic.
But this one specifically is natural, which is significant in a way because it doesn't require some of the. It's not a medical. It's not a drug that was developed.
Yeah, I mean, it hasn't had to go through the drug development process, although we started that way. So the original discovery was made by Professors John Stamford and Professor John Grange, who were microbiologists at University College Hospital, London. And they were trying to work on an adjuvant, and you've heard the term immunoadjuvant, which is what tries to improve the effect of a vaccine. Most vaccines don't work properly without an adjuvant. And the vaccine for tuberculosis is called BCG, Bacille Calmette Guerin. It is a kind of adjuvant in itself, but it still needs an external adjuvant if you give it to anybody over the age of 3. So it's a good TB vaccine under 3. It's not so good over 3. But there are some parts of the world where you give it to adult patients, you get terrific responses. So it was noted that in Uganda, on the shores of a lake called Kyoga, there were these 11 villages. 10 of them were cereal farmers, 11 were cattle farmers, 1 was cattle farmer. And the 10 cereal farmers did not respond well. The cattle farmers responded fabulously to the BCG.
How come?
And they thought, well, hang on, there's something odd going on here. There must be an immunoadjuvant going on. And the only difference was that these guys took their cattle to the water twice a day through the mud. So they cultured the mud and everything else. And they found this Mycobacterium vaccae for cow, which we've reclassified as a Mycolysibacterium kyogaense after
a lot of words.
Kyoga. Yeah. Because it's not actually vaccae. But anyway, it didn't matter. The point is that they thought they'd resolve the issue of tuberculosis, which, by the way, is a biblical disease, and it still hasn't gone away. I mean, TB is on the increase.
A recent outbreak in the U.S.
Yeah, and actually, the UK is probably the leading. It's not a great accolade, but it's the leading center for tuberculosis in Europe because we've got the most cases, and these are drug-resistant versions.
Mm-hmm.
So that set up the studies for TB, and there were lots of other TBs that have been done over the years demonstrating that vaccae and the second organism, Mycolysibacterium obuense, that we used, and it was then used orally, has a dramatic effect on helping the quality of life signals in tuberculosis patients. This was a study that was done in Asia.
You actually have an extensive background in cancer and immunology, so I'd love for you to share more. I've heard that you basically invented laparoscopic surgery. You've been studying this for ages. You've been in the field for ages. Tell me more about your history with cancer.
Well, thank you. I'm not sure I invented laparoscopic surgery, but I was one of the people that adapted it to modern surgery. So I wouldn't want to claim that. I would love to, but it's just, humility. The cancer thing to me is much more important. It's always been what's driven me. I want to get rid of cancer, give it a bloody nose and knock it down before I go.
Yeah, absolutely.
And one of the opportunities that arose has been this work using the mycolysibacteria. And I suppose it started originally work with tuberculosis, but we moved into cancer very quickly. And there was a phase 3 lung cancer study. That's several hundred patients were admitted into this study. So we're talking about a serious piece of work done at the Royal Marsden Hospital in London, like the Sloan Kettering in New York. It's a great center. And what we found, although the results from using the precursor to IMI, Amvaki, that the results weren't as fantastic as we thought in terms of curing the cancer, because we didn't know about the way that you have to use immunotherapy in cancer. We were very naive. We thought it was a drug, just like a chemical drug that you would use. What we did find in those patients who were injected was a fantastic quality of life effect. And that was published by Dr. Mary O'Brien, who ran the study subsequently. And that quality of life included things like coping, reduced stress, reduced anxiety, and things like not losing weight in the same way. The other comments we've had from experience with other cancer trials that we've done, and we've done quite a few, is that the patients would tell you, I should be getting coughs and colds because I'm on chemotherapy, but all my relatives got coughs and colds last winter, and I was absolutely fine. So this kind of bystander immunity effect was really interesting. But the stress effect was important. That's what led Professor Chris Lowry to start investigating. He's a neuroscientist at Boulder, and Professor Stefan Reber at Ulm in Germany, to start investigating the neuro effects of these mycobacteria. So we then carried on and we did further studies, but we still didn't fully understand the best way of harnessing the power to try and actually hit the cancer hard, until we realized that we were going about things the wrong way. And so in our experimental developments, and you have a paper I know from as long ago as 2017, we're going to update that big time. Yeah, we're going to update that
What
big
did we
time.
learn from this paper? All I see are charts. You might have to debunk this for me.
Listen, that's transcriptome data, and you need a PhD in immunology to work that out. And I don't have a PhD in immunology, but I mean I can work it out for you. But I don't think I think it's a bit too sophisticated for everyday purpose. And it's got even more complicated because the new single-cell transcriptome data, which we have from currently being produced in Texas, it provides 60,000 data points. So one of the reasons I say I don't want to go into the details is the data, every cell produces 60,000 bits of data, and you just get swamped. I mean, trying to explain it without artificial intelligence and the supercomputer is impossible. But we are on the case, and we'll get fantastic data exactly how IMI works, because we've used it on human cells. We don't do animal experimentation. We've done it on human cells, and we see the effect that these transcriptome studies work on the genes, and gives us a lot more information of these human cells.
So that's really interesting, because we're talking on a cellular level about improving cancer response, right? Or in other ways, like, can we fight cancer with natural therapies? You mentioned to me before that if you're taking an immunoadjuvant like IMI, Mycolici bacteria, it actually has the ability to repolarize corrupted cells. So when you have cancer, you have corrupted cells.
What
happens when you repolarize that cell?
It's actually modulation rather than is the term, rather than adjuvant, but
right.
it's repolarization is a good term, and then particularly the modulation to change the effect of the cell. So what happens, cancer is really, really smart. It wants to live. It's a real pig to deal with. It's stubborn. It steals your mitochondria. It steals your food. It's a badass, as you might say. And so what happens is the immune system tries to attack it. And I use a dog analogy. You know, if a burglar comes to your house and tries to get in over the high wall in your garden, and you've got a Rottweiler there, the Rottweiler will just go for the burglar and attack him. It doesn't have to decide whether he's a burglar, or anybody who goes over that wall is fair game.
Yeah.
And that's the macrophage. That is what happens normally in a healthy person. The minute the cancer cell raises its head, and we produce cancer cells, probably 100,000 cancer cells a day is I've seen quoted, but enough cancer cells every day, but we clean them up because the immune system says, hang on a minute, this is not good. The Rottweiler goes in, attacks them, eats them up. The macrophage eats them. It's a big eater, and gets rid of them. Now, cancer then gets quite smart. So what the burglar does is he throws a piece of meat with a bit of dope, something to poison the dog.
Poison meat.
Poison meat over the wall. The dog eats the poison meat and goes to sleep or dies. Cancer then climbs over the wall, and it's got in. It's got to start. And that's exactly what happens. And so you need something to wake up that Rottweiler, because he's asleep or he's dead.
Mm-hmm.
And so we see, when you look at the cancer, a piece of tissue that's got cancer in it in under the microscope, you will see the cancer cells, but you'll also see immune cells scattered throughout, and they're just sitting there. They're fast asleep. They have been exhausted. They're tired. They don't do anything. So our idea is to say, well, okay, hang on, let's wake these guys up. Let's get them moving. And it's not just us. Everybody else in the business is trying to turn these cells on so that they become hot again and active and attack the cancer. And so what we've demonstrated very clearly, and that article has done, is that if you put the mycolysis bacteria, and even if you put IMI, we've proved that IMI does that, and you put it onto these exhausted, polarized cells that have been corrupted by the cancer, it wakes them up.
Interesting.
And it does. And it's extraordinary. And if you then take those cells and put them onto another cancer, it will kill the cancer. So we know we've got something extraordinarily powerful. What's even more interesting is that it's not a simple repolarization, going back to yin and yang. They're called M1 or M2 macrophages or DCs, dendritic cells. What happens is you get a phenotype, a pattern of behavior that is neither one nor the other, but is very smart. So it's a bit like having a Rottweiler and a Chihuahua. The Rottweiler's put to sleep, so basically there's just a Chihuahua going on. We don't really want just a Chihuahua sorting out a cancer and a 600-pound burglar. Chihuahua not going to do very well. But this repolarization, if you like, modulation that occurs with particularly IMI, is a blend between the two. Smartness of the Chihuahua and the power of the Rottweiler.
So
brilliant.
That's so interesting. So you're telling me that we have original cells. They're like Rottweilers. They get rendered pretty much useless. Sorry, Chihuahuas. But they get rendered useless. They turn into Chihuahuas when they are attacked by cancer cells.
Yeah.
And as a result of an immunomodulator, like something like IMI, you can actually wake up the cells and reprogram them so that they can respond appropriately to cancer cells.
Exactly.
Kind of like a German Shepherd.
Yeah, well, the German Shepherd's smart. He's more of a different kind of adaptive immunity. I use that in a video that what we want to do. So obviously we think IMI is fantastic, because particularly of the effect it has on stress and coping behavior in ordinary people. And because of that, for ordinary people, of which there's a huge problem with stress, and by the way, long-term stress is not a good idea for all sorts of illnesses, and chronic inflammatory changes occur, which again are bad for you. Mm-hmm. So IMI, we're using that for stress and for helping people deal with chronic inflammation, because we want to keep it at a cost that is viable for most people, $1 a day. Most people can manage $1 a day. Our problem with developing the cancer treatment is that we'd already spent close to $100 million, and we still needed to produce another 200 or 300 million, which is very hard work. We're not a big pharma. Big pharma will spend $2 billion to get a new cancer drug to market, and you'll charge you $100,000, $150,000 for a course. We don't want that. What we do want to do is, when we get funding and when we get the new data that we are looking at the moment, is to extract cells from the patients, remodulate, repolarize them, and then put them back to attack the cancer. And we think that's got a better than even chance of having a really good effect. And that's the way modern cancer treatment, we think, is going to go.
That's so powerful, because in many times cancer feels very hopeless, right? Yeah. So you're telling me that we have not only a way to fight cancer, fight chronic conditions that do attack the immune system. Could be everything from Lyme disease to Hashimoto's. But not only is there a way to do this, but it's with a natural therapy.
Yeah, and the proof of that is a condition called malignant melanoma. Now, the poster child for that actually is the late President Jimmy Carter.
Right.
Right?
Oh, yes, that's
Yeah.
right.
He had malignant melanoma, and he had brain deposits from malignant melanoma in 2016. I'm not letting any secrets out. This is public knowledge, right? So 2016, which is 8 years ago plus, he had, and he only died earlier this year. So he survived 8 years with brain metastases. When I was in full-time practice, if somebody had malignant melanoma brain metastases, if they lasted 6 weeks, it was a miracle.
So how did Jimmy Carter live so long with cancer?
Because he responded to the checkpoint inhibitors, to immunotherapy. And he was able to do that because he was one of the lucky 50% that respond incredibly well to these checkpoint inhibitors. They're very expensive, but when it works, my goodness, it's fantastic. So they get a 50% virtual cure rate, however advanced the cancer is. The problem is if you turn that on its head, 50% don't respond. Now, listen, in my day, you were lucky to get 4 to 8% 5-year survival from advanced melanoma. Now you're talking 50%. It's fantastic, right? But still, 50% to go. So what a group did was take stool and do a fecal transplant from the responders to the non-responders. The 50% who responded into the 50% who didn't respond. 30% of them then responded. Another group in Israel took stool from healthy people, put them into the non-responders, and they got a 50% response rate.
Incredible.
It is incredible. And what that tells you is the microbiome, because that's what the fecal transplant does, is fantastically important in terms of immunotherapy. And that interface between the microbiome and the immune system is just everything. And that's the bit where IMI and the mycolicibacteria sit. It's that interface between the microbiome and the immune system, and that relationship between the two that makes the magic happen.
I mean, it's super interesting. You know, we've talked a lot about this concept of farming your microbiome. You produce this garden, right? And then where does the immune system come in? It's this farmer. He comes in to take the fruit.
Yeah, that's a great analogy, isn't it? So you do have to, I mean, we've discussed this, and whether you're a good gardener or a good farmer, it doesn't matter, but you need thousands and thousands of different plants as a gardener. You need lots of different variety as a farmer. But what the farmer does, and the gardeners, they look after the microbiome, if you like, their farm, as we should. And you give it fertilizer and food. So there's your prebiotics and your, fiber.
Probiotics.
Yeah, and you put seeds in, the probiotics, whether it's lactobacilli, one sort or another, but your juicing, your interaction with your environment, your family, you've got to have a lot of interaction with the environment, because you need to expand your microbiome to get diversity. The secret is diversity. It's not, 5, 6, 10 perfect bacteria that do the job. It's the 40,000 that you really want. I mean, that's a lot of bacteria. So that what that happens is they farm it, they harvest the crop, because the microbiome, the crop, produces all sorts of wonderful things, whether it's hormones, whether it's special chemicals that we can't produce in our body, and there are an awful lot of them. All of that stuff then goes back into the body. But that link with the immune system is absolutely extraordinary. So you need to make sure that farmed microbiome is triggering a good response in your immune system, all of which goes through the gut. The gut is the biggest immune organ in the body, so it's the right place to be. And then the immune system can work properly. And your immune system protects you. It deals with repair. It rebuilds you when it's working properly. And it knocks out all sorts of bad things, whether it's cancer or, viruses or whatever. It's a very important component of life.
Very interesting. I think if I were to even rewrite this episode, I would say the story of IMI started in an immunotherapy for cancer, and now it's available to everyone. But if we were to boil it down, it's really a truly natural remedy that has helped so many people survive cancer, improve their quality of life and to all other chronic conditions.
Yeah, whether it's helped them survive directly, that's a big term. We would love that to be the case, but we haven't proved it.
Right.
And you know me, I like facts and
For sure.
proved evidence base. So, however, the effect in terms of mood and anxiety and coping behavior and resilience and maintaining weight is not in doubt. So those patients with cancer do benefit. There's no question about that, and that's good. We'll take it. It's a start.
Yeah. So okay,
There's a long way to go.
I am a budding cancer student. Very new to the space. So my understanding is, when you have cancer, it's like this burglar trying to infiltrate your house. They're coming in and they're going to do bad things to my cells or to my home, and when we have corrupted cells, we don't function properly.
Correct.
Right? Cancer steals energy from our healthy cells, and you kind of waste away because it grows its own mind and it's growing its own. It's like a satellite parasite.
It's a parasite. That's a very good analogy. It's an obligate parasite.
Right. So with this parasite, it's stealing our energy. It's messing up our current system. And with an immunomodulator, like something like IMI, you can actually reduce the harmful effects that these cancer cells.
That's what we're trying to do and to prove. The problem is getting it to the right location.
Mm-hmm.
And that's why we think that putting it directly into a particular cancer, so-called intralesional therapy, where we put it into the tumor directly, is the way to go forward. And we're working on trying to get a study done for that, because the evidence, as I've suggested, even just from that paper, but never mind the new papers, and I have to be a bit careful because we're going to publish new data, and we can't release information until we've published it.
Okay.
But it is super exciting. And if we can do that, we'll have done something really quite wonderful.
Yeah, pretty significant. I mean, for many people, like 50% of us will suffer from cancer. So
Correct.
Either yourself or myself, we know someone or their family member who's been affected.
Yes.
Would you recommend IMI for them to take IMI as a supplement that can help relieve the symptoms and help improve their quality of life while they battle cancer?
I mean, if you look at the data from that lung cancer study, the answer is yes.
Mm-hmm.
It does make a difference to the quality of life, however you measure it. So, and there is evidence that if you improve the quality of life, you improve survival by at least 10%. This is from the European Cancer Consortium.
Right.
So I didn't just make that up. It's been published.
Yes.
So quality of life is really important and can affect survival.
So we can't say today, we can't promise the survival if a cancer patient's taking it, but we can produce a correlated effect, which is if your quality of life is better, you can cope with stress better, you can respond with more resilience, then you have a significantly higher chance of surviving cancer.
Correct. And if we can find enough funding and get the work done, boy, we
just need $200-300 million more dollars.
Well, some kind soul, I'm sure, has got it stashed away somewhere, but that's a lot of money.
Yes. So, when we talked about this innate and adaptive immunity on the previous episode, you brought it down even to a molecular level. You said, okay, you're either born with innate immunity or you can learn adaptive immunity.
Correct.
So for this immunomodulating effect that we're talking about for adoptive cell therapy, is that considered innate? Are you born with that or are you learning that? Is it adaptive?
Yes, no, it's innate. You're using the innate cells, that's the macrophage and the myeloid series, primarily to induce the response that you want. There's a particular chemical which is really useful that then brings in other cells like the T lymphocyte and other things that will then bring in innate adaptive immunity. Adaptive immunity is the learned immunity after a vaccine, for example. If you get a vaccine, you learn how to deal with it. Innate immunity is the Rottweiler in the garden. He'll just go for anything, right? Mm-hmm. But we use innate immunity, and we can train innate immunity to our benefit. Even a Rottweiler can be trained, okay?
Amazing.
So that's really what we're after. We want to retrain, remodulate, repolarize, whatever you want to call it, that Rottweiler, so that he does what he's supposed to do without being shut down by the cancer, which is smart.
Right. And you defined macrophage in the previous episode. If you can do it one more time here.
Yeah, the macrophage means a big eater, macro, big, and phage is eat.
Mm-hmm.
And the macrophage is, I mean, there isn't just one macrophage. I think I told you there's dozens and dozens of them. But the thing about the macrophage, first of all, it's a big eater. So if it sees a bacterium or a virus, it'll just go and gobble it up, and it then kills it. If it sees a senescent cell that is not doing very well, it will go and clear it. Even if it's like a zombie cell, that is there, its job is to go and get rid of those cells. Right. And it's very good at it, right? And then it it sends out signals so that other cells come in and do the repair, particularly in that situation.
Yeah.
So in terms of senescence, having a good macrophage is great, because then you keep the repair process and the healing process is really good. If the healing process is damaged, right, then you start getting old. And the key thing for that is HIV, human immunodeficiency virus infection. The clue's in the name. Immunodeficiency means that these cells can't do their job. And certainly before retroviral medicines came in, I mean, I'm so old that I saw some of the very first cases in the world back in the 80s. You really see this aging process happening in front of your eyes. These poor men and women would just melt away in front of you. Like with advanced cancers, I've seen so many advanced cancers where the patient just melts in front of your eyes.
I know, it's terrible.
And it's awful, horrible.
It's, this is such an interesting point around protecting or really being able to build resilience in macrophage, because it's not a cell. It's not a cell type that has been talked about recently, it's not a cell type that is quite popular to talk about. In the longevity space, people are talking about zombie cells. They're saying, well, we have all these dead senescent cells and we got to clear them and get rid of them. But who's actually doing that job?
Well, it'll be essentially the macrophage. That's its job. The macrophage, every organ system has got its own version of the macrophage, right? And they have different names. So basically, they're the same old thing. They're the dustmen.
The detox people, the detox buddies.
Yeah, they go around and they take, they clear up the mess. And more to the point, they repair the damage. And that's what the macrophage is supposed to do. And it's a big eater because it eats the stuff and chews it away. Now, you may think, well, that's not going to do very much. But actually, if there are millions of them, it's amazing what they can do, isn't it?
Yeah. So from these papers, what are the most significant takeaways that we can take from them? Like, we can't say, well, today we know that we're having a positive effect on patients with cancer and their quality of life, but we can't say that we're curing cancer, but we can alleviate some of the suppression effects on the immune system from cancer.
Well, and you're absolutely right. We certainly can't say we're curing cancer using this, although it's very interesting the anecdotal cases that we have of long-term survivors with pancreatic cancer are quite extraordinary, but we haven't been able to prove it in a formal clinical trial, which is the gold standard, right?
Yeah, absolutely.
But the evidence is there. I mean, I've told you this before. If you throw 100 tennis balls off the top of the Empire State Building and 3 of them float, you've got to explain that, right? Something's going on.
Yeah.
So all of that is really important. And we think that the data that we're seeing, the data that we've got from 2017, that paper, and the data that we're seeing now, and we had a big delay because of COVID and all that stuff, the data we're seeing today, and we hope to publish as soon as we can, and interact with the NIH and getting some studies done, further studies done, is mind-boggling. I mean, it is just amazing data. And it demonstrates that not only does it have a therapeutic action, these cells that are repolarized will kill the cancer, but they will also prevent a future cancer. Now, that's a really big claim.
That's a really big claim. That's preventing the growth of more tumors.
Yeah.
Apoptosis.
Wouldn't it be great if that happened? That's what we're hoping for. But until we see the data and until we, crunch it, that is a huge amount of data. You need a big supercomputer and AI and all that to even get started. And that's going to be really exciting. If it's half true, they'll do.
Yeah. What are common quality of life signals for everyone listening?
Well, they felt better. They didn't feel fog and sick. They had less stress and anxiety. They could cope better with their situation. They didn't lose weight. They had an appetite. And generally they resisted other infections, of which they're very prone.
Right.
So these are all quality of life signals that just make life a bit better. The same happened because the vacci was then, it was thought that this was a potential cure for lung cancer, and a big phase 3 study was done, which demonstrated not a great response in the lung cancer as such, because we didn't understand how to use it properly. We were still trying to treat it as if it was some kind of chemical drug, right? Which immunotherapy is not. It's a huge mistake. So what we did see, and what was published by Professor Mary O'Brien, who was the oncologist at the Royal Marsden in London, was this quality of life signal. The patients who walked in, you could tell immediately. You couldn't blind the patients in what's called a double-blind clinical trial, because the minute they walked in, the nurses, everybody, oh, you must be on IMI, or you must be on vacci, because, they've got a smile, they were coping. It was amazing.
That's kind of wild, right? That you're saying we've discovered this bacterial strain that improved the quality of life and also the mood and the coping resilience of tuberculosis patients. And you saw similar corresponding responses in patients with lung cancer.
Exactly. And we've seen it with pancreatic cancer, because we then used Obuensi in a 110 patient trial with pancreatic cancer. And that's a horrible cancer.
So it's not just lung cancer, it's pancreatic cancer.
It's across the board. Melanoma, our best results in the clinical trials, funnily enough, were in melanoma. We've had some very long-term survivors in melanoma treated with Obuensi, which is wonderful.
This is kind of game-changing for anyone with a family member suffering from cancer, because your quality of life is severely diminished, right?
Yeah.
If there's a way to improve mood or stress or resilience in coping, it's not going to solve the cancer, but it's certainly going to improve the quality of life that you have around it.
It may improve survival, because we know that in cancer cases, if you've got a good quality of life signal, you can improve the survival by up to 10%. And this is published by the European...
This is published data.
Yeah, published data. I don't make data up.
Yeah.
So it is published data. So if you've got a good quality of life response, why wouldn't you do it? In Japan, 85% of the patients with cancer take mushroom extracts.
Okay.
Right? And whether it's turkey tail mushroom or any of these other ones that are now available. And thereafter, the beta-glucans. It's the beta-glucans that acts as a nice immunoadjuvant.
Yeah.
Beta-glucans is an adjuvant. And it seems to have they like it. They feel better on it and what have you. In the same way, you could take beta-glucans, but we think the signal is much, much better with IMI. Because there's a therapeutic effect, potentially, in addition to just, making you feel better.
That's incredible, because anyone suffering it's a no-brainer. You ask someone, do you want to feel better while you're recovering from cancer? But it also may, we won't know. We have emerging data soon. It may inhibit the growth of future cancer.
Correct. We
That's
know
crazy.
that in the single cell transcriptome data, which is really complicated stuff, where we look at the DNA signals from tumor cells, not tumor cells, from the immune cells. The problem is every single immune cell produces 60,000 data points.
Yeah, that's a lot.
And that's we've got to chunter through all that lot. But it's very accurate data, and we've already had some data in that 2017 paper, which is incredibly exciting. The data from our latest work is even more exciting. I have to be careful not to say too much, because otherwise we won't be allowed to publish it.
What we learned is that you can repolarize corrupt cells, and you can speak to that when we have more data, but that's game-changing, because primarily when cancer is corrupting the cells in your body, you don't have a response. If you can repolarize and train your immune cells to respond and to attack cancer, that's significant. That's huge.
The dog up.
Yeah.
As I say, unfortunately, IMM 101, we haven't been able to develop the Obuensi because they run out of money. And the reason for that, because as I say, we'd spent 100 million, and I did not want to be having to charge somebody $100,000 for a 6-month course of treatment when we discovered that actually IMI works really well orally by mouth. We've got data, by the way, on diabetes. We've published that paper recently. It works orally because the gut is a fantastic center for immunotherapy.
And you also mentioned that there is reason to believe that M. aurum aogoshima is actually more effective than oboense, the sister strain.
We think it is, and we can do it for $1 a day, whereas if we did oboense, it'd be $100,000 for a course. So $1 a day, anybody can do it. And as an old doctor, I went into medicine a long, long time ago. 1970, I walked into medical school. So imagine how old I am. And the idea was to benefit my fellow man, not to make money out of their misery. And $1 a day, most people can handle.
Well, I mean, that's really exciting and can't wait for more of our research to emerge. But as for now, I think we can safely say that we are encouraging people with chronic conditions, could be anything from Lyme disease, tuberculosis, Hashimoto's, those suffering from cancer, to try IMI as a resource. It's a natural therapy. It's a bacterial strain, a very friendly bacterial strain that can help with the immunomodulating effects and help protect you from those.
And it does what it says on the box. It's not hype. It's not based on hearsay. A lot of supplements are just nonsense. There's no evidence that they help. We have, $100 million worth of data,
I mean, it's hard because nowadays there's just someone trying to sell you something for everything. And you keep saying serious science, and it does sound quite serious.
It's got to be serious.
Yeah. So, well, I'm excited. We'll stay tuned for more research, but we're going to wrap up today's episode.
Thank you very much.
See you soon.
Bye.